Hey everyone, this is Dr. Medicine RX, and in today's video we'll be discussing about the structure of Mycobacterium leprae, which is a very complex topic. I've tried to simplify this to the best of my abilities, and I've also tried to include simpler sketches for the same. So Mycobacterium leprae is a rod-shaped bacterium which has a length of 1 to 8 microns, a diameter of 0.3 microns. It has parallel sides and rounded ends, and it is strongly acid-fast on Ziehl-Neelsen stain. Acid-fast means that the bacterium it is able to resist decolorization when we add an acid due to the presence of large amounts of mycolic acids in its cell wall. Next is the globus phenomenon. It manifests where the environment is favorable for rapid multiplication, therefore in infectious cases or the cases that are towards the end of the spectrum, that is the cases of borderline lepromatous and lepromatous leprosy. This is basically the spherical conglomeration of rods where inside the cells that are the macrophages, the M. leprae, they multiply unrestricted and they get arranged in parallel arrays due to the presence of surface lipids. This gives a bundle of cigar appearance which is known as the globus phenomenon. Coming to characteristics of the M. leprae, these are non-motile, non-sporing, microaerophilic and acid-fast bacilli. Next, coming to morphology, on electron microscopy, we see that it has four main structures. First is the capsule, the second is the cell wall, the third is the cell membrane, the fourth is the cytoplasm. These are from outside to inside. In this rather simpler sketch, the number one bluish structure is the capsule. Number two are the mycolic acids, which are linked to number three, the hexagonal structures, which is the arabinine, which is again linked to galactan, which is the bluish structure, which is further linked to peptidoglycan, which are the greenish structures, and this is followed by the cell membrane, which is number 6. Next, coming to capsule, it contains bacterial lipids which are found in large amounts in infected tissues. So there are two main types of bacterial lipids. The first is the thiocerol dimycoserosate, which is an apolar substance and it has a passive protective function. And the second is the phenolic glycolipid 1, which is specific for M. leprae and it is active serologically and it is found in abundance in infected tissues. It is produced locally and it can constitute the characteristic foamy macrophages of the lepromatous leprosy patients, and it is highly immunogenic and it generates IgM antibodies, and ELISA is used to detect these specific anti-PGL1 antibodies. In addition to these lipids, capsule can also contain lipoglycans such as the lipomannan, lipoarabinomannan and phosphatidylinositol monocytes. These are usually found in the plasma membrane, but they can also be present in the capsule. Next is the cell wall. This is the outer coat and it protects the bacterium from the external environment and it also gives a definite shape to the bacilli. It has outer electrolucent layer and an inner electron dense layer. So this outer electrolucent layer further has outer and inner leaflets. The outer leaflet, it is formed by the mycolic acids of the trihalose monomycolates, mycoserosic acids of thiocerol dimycoserosates and phenolic glycolipids. The inner leaflet, it is formed by the mycolic acids that are linked to terminals of arabinine chain. Then the inner electron dense layer is formed by the peptidoglycan, galactan and arabinine. So basically cell wall is a covalently linked structure formed by the peptidoglycan, arabinogalactan and mycolic acid complex. So this peptidoglycan, it forms the core of the cell wall and it has alternating N-acetylglucosamine and N-glycolyl muramate. Next is the cell membrane. It contains transport proteins, basically proteins which control the active and passive transport of substances across the cell. It also contains the enzymes that are required for the synthesis of the cell wall and capsular lipids, and it also contains phospholipids which are characteristic of membrane of cultivable mycobacteria. There are two major polypeptides found in the cell membrane. The first is the major membrane protein 1, MMP1, which is a 35 kilodalton protein and it is a serologically active component. And the second is the MMP2 protein, the major membrane protein 2, which is a 380 kilodalton protein. Next is the cytoplasm. It has three major proteins, a doublet at 28 kilodaltons weight, a protein with 17 kDa molecular weight and a heat shock protein. Another 65 kilodalton antigen is also found in an extensively degraded form of M. leprae preparations. Thank you. Subscribe for more.